Hello, dear students. Once again, you are most welcome into the Biology Pathshala by Oja sir. And in Standard 12th, chapter number 5, Heredity and Variation, we are going ahead with one more concept, that is rediscovery of the Mendelian genetics. With the advanced technology and advancement into the research in the science and technology, there are few basic fundamental acceptance which were not proved by Mendel has been rediscovered. All the Mendelian experiments were rediscovered. So how it has happened, let us see. Uh, the main conceptual theory is the chromosomal theory of inheritance, what we are going to understand now. Due to three main reasons, which is given below, entire work of Mendel, that is in 1865, was remain hidden. The first ever reason is uh, at that time, in that era, communication is or was not that easy. So uneasy communication in that era is the basic reason why Mendel was not becoming famous at that era. Number two, he was unable to explain any logic and couldn't prove anything about the factor which he told that factors are responsible for inheritance, but he could not be able to make other understand the mechanism how the factor is inheriting from one generation to the other. And due to various differences in a particular characters, his mates couldn't accepted his hypothesis that factors are not mixing with each other. As we know that there are two alleles, that is two factors with one gene which is responsible for inheritance of a unit character. So two factors or two alleles which are not mixing with each other, the other scientists were not digested it and could not be able to accept it. And third reason, the cumulative analysis what Mendel has done, or mathematical point of view of Mendel. He applied mathematical rules, and it was new in that era and nobody could have understand that phenomena, right? So that's why these are the three main reason why Mendel was not accepted by all other scientists or the society. And rediscovery of Mendel's work is made by three scientists in 1900 later on, after the death even of the Mendel, right? First is Correns. He has experimented on maize plant. Second was Hugo de Vries, and he experimented on evening primrose. Third was Schermark, and he has experimented on various other flowering plant with the same Mendelian principles and Mendelian experiments. So that's the key factor, rediscovery of those three main principles of Mendel, which has been done by these three chief scientists, Correns, de Vries, and Sherman. Now, Walter and Walter Shatton and Theodore Boveri in 1902, they have given, they have observed and given one basic principle, right? And that is what, after the discovery of science and technology, after the discoveries of the various types of microscope including high power microscope, so scientists are now being observed internal behavior of the cells, internal structure of the cells, internal mechanism of the cell as well, living, that is into living cells. So that is how they have observed cell division process and they have tried to evaluate that how the mechanism of forwarding these genetic material from one cell to the other, that is one generation to the other. So that's the experiment of Sutton and Bowry, what we are about to understand. It's known now that due to unavailability of technology and microscopy, Mendel was unable to prove his work, but Sutton and Bowry has studied cell division and interaction of chromosomes in nucleus. They have compared behavior of chromosome with the behavior of factor, that is gene of Mendel. So whatsoever observation have Mendel done, it has explained it with factor or gene. So behavior of factor, law of the genetics which have been given by Mendel was to the factor, dominancy, segregation, independent assortment, whatsoever rules what we have studied till now was given by Mendel with consideration of factors. But he could not be able to know the chemical structure or chemical nature, that is behavior of the same factor, that what these factors are made up of. So to understand that, to know that whether factor are what, Sutton and Bowry has tried to study cell division, tried to see the interaction of genetic material, and then they have seen the chromosome in nucleus. They have studied complete behavior of chromosome, and they have compared the behavior of chromosome with the behavior of factor of the Mendel, the principles of the principle of Mendel's experiments. They have compared it with the behavior of the chromosomes. They also have proved that the heredity is maintained by genes which are located on chromosome. So Mendel have given factor or gene name, and where the gene or factors are lying, that was proven by Sutton and Bowry. They said that they are located on chromosome, right? And these genes are forwarded to next generation in the form of chromosome. So chromosome are group of gene in the other sense. So genes are located on chromosomes. So each chromosome is containing so many genes, so many factors together. So definitely they are in pair, as we know that chromosomes are in pair in diploid organisms, right? One of each factor is dominant while other is recessive. So that's the first law of Mendel, Mendelian genetics, that either of it is dominant and the other is recessive. So that way, first law is applicable. Then assortment. At the time of gamete formation, again they are separated. That is demonstrated here into the diagram. If you observe in G1 phase, there are four different type of chromosomes which are given, right? One is the pair is uh, yellow and red. Second is orange and green. Fine. In G2 phase, if you see, it is doubled. Why? Because during S phase, uh, the entire genetic material is getting doubled, right? Replication is the process by which it is getting Xerox copy of each other. So here, material is getting doubled in G2 phase. So they are becoming bivalent in nature. Two chromosomes, one original, one is duplicate of each. It is into the pair, which is known as homologous chromosome. So those homologous chromosomes are getting into the pair, which is into meiosis 1 anaphase. Into the pair, they are getting separated. One is going toward north pole, and two pairs are going towards north pole, and two pairs are going towards the south pole. If you clearly see into the diagram, then you could have noticed it. Here there is no separation of chromosome from the centromere region. That is the uh, that's the speciality of meiosis, right? Meiosis 1. In anaphase of meiosis 1, there is no separation of chromosome. There is no depart of the two sister chromatid at the place of the centromere. That's the speciality. During meiosis 2, they are getting separated, right? It is just copy of mitosis. And later on, when look after meiosis 1, they are getting separated into two cells, and after meiosis 2, they are getting again, each one of them is getting separated into two germ cells. So total number of germ cells are 4. Now if you see G1 phase and germ cell, the pairing of chromosome red and yellow, they are getting together, and orange and green, they are getting together, but they are separated from each other. That is law which is proving segregation process, the second one, right? Our second law of segregation is proved here. The next characteristic feature, they have explained that pairing of chromosome and their assortment is responsible for assortment of factors, which is said by Mendel. Mendel has said that factors are assorted independently, but how? The mechanism is shown by Sutton and Boveri. And this entire set of the whatsoever observation they have made, they have known, they have given the name as chromosomal theory of inheritance. So that's what the story of Sutton and Boveri's chromosomal theory of inheritance, right? Now they have compared the behavior of gene and behavior of chromosome altogether. Now what they have got by their comparison, what they have observed during the comparison, the first comparison what they have made is, number one, gene are always in pair, and if you see chromosomes, they are also in pair. So gene, the unit factor which has been the concept has been given by Mendel, and concept which they have proposed, Sutton and Boveri, they have proposed chromosome. Both are matching, right? Both are into the pairs. Now, if I talk about genes, second characteristic feature, they means allele are assorted from each other at the time of gametogenesis, that is gamete formation, and only one is forwarded to each gamete. One allele is going into one gamete. Similarly, if I talk about chromosome, the second point is they means chromosomes are also assorted the same way, that only one chromosome is forwarded to each gamete at the time of the gametogenesis. Third behavior of gene is one pair of particular gene is independently assorted or forwarded when they are forwarded into the next cell, next generation cell. And if I talk about chromosomes, one pair of different chromosome is assorted independently to each other when forwarded. We have 23 pair. So first number pair is not dependent on 23 number pair for forwarding it into the next generation, into the gamete. So that's what important comparison what we have, what we can think about. Uh, it is matching with the genes or factor which has been explained by Mendel. So that's how importantly they have proposed the chromosomal uh, theory of inheritance. If you see the process of chromosomal theory of inheritance, third rule, independent assortment of the chromosome, figure number 5.9 of your textbook, there is two possibilities given. Possibility number one, possibility number two. If you think about the possibility number one, One long orange and short green chromosome, which is appeared in first diagram, that is in meiosis 1 anaphase, and long yellow and red short chromosome are at the same pole, right? So in first diagram, that is meiosis 1 anaphase, uh, yellow and red, and the other pair is orange and green. Now they, when they are separated at the time of making of germ cell, that is gametogenesis, they are placed as it is, they are forwarded as it is. So that's the one possibility. And the other possibility, if you see, one long orange and short red, and the other long yellow and short green, they are interchanged, if you see, right? Previously yellow was paired with red, here orange is paired with the red. Previously orange was paired with green, here yellow is paired with the green. So that's why independent assortment is seen here, what we can observe clear cut here into the case of possibility to meiosis 1 anaphase. If you see, similarly, when they makes the germ cells, yellow pairing with the green one, and orange is pairing with the red one into the germ cells. So uh, green, uh, which was actually in possibility one with the orange one, is here right now with the yellow one. So they are showing the independent assortment of the chromosome in this way. So all the major three law which has been denoted by specifically to the Mendelian experiment are applicable equally to the law of the theory of the inheritance by chromosomes. So by chromosomes, they all rules are followed, right? Now, based on chromosomal theory of inheritance of the Sutton and Bovary, next experimental proof was given by Thomas Hunt Morgan and his colleague to prove them, right? So experiment was done by Thomas Hunt Morgan and their colleague. Why? To prove the inheritance of Sutton and Bovary, that is chromosomal theory. Now they presented the theory of linkage and recombination by their experiment. So theory of Thomas Hunt Morgan is named as principle of linkage and recombination. What it is, how it is, that's what we are studying later on into the next lecture. But today we are getting introduction of the theory. Now Thomas Hunt Morgan has experimented on Drosophila melanogaster, that is fruit fly. As Mendel has selected pea plant, he has selected Drosophila with the similar manner of the easiness to grow. Right? First character why he has selected Drosophila melanogaster is easy to culture. Cultivation is very easy. Easy to maintain them. Right? Second, many offspring they can produce, and clear difference in male and female. If you see the diagram which is shown there above, male are small one, female are the large one. So you can differentiate between male and female very easily. That's why it is easy to breed them, right? So clear cut difference between male and female. Easy to cross, self cross is very easy, and hybridization also is very easy with those kind of Drosophila melanogaster. Fourth, very important, very short lifespan. And fifth, limited characteristic feature and easy to observable contrasting character they can show. Height, weight, color, type of some behavior, eye color, etc. So that's the very comfortable for the experiment. That's why Morgan has selected Drosophila melanogaster for his experiments, right? Now, linkage is what? So if you see the definition which is given here, when genes are located very close to each other in a group, they are inherited as a single unit factor because they are so closed, and this phenomena is known as linkage. Now Sutton has proposed principle of linkage in the following manner: number of group of gene and or number of group of chromosomes are same, but he could not be able to mention them or prove them. But linkage principle which is given by Morgan is slightly different. How? Look at the theory given here. So genes of homologous parents transfer in one gametes and remain together, but it is reversed in heterologous parent. Linkage or linked gene which is into the group. So linked gene group are located on same chromosome only, and distance between them controls a recombination. Fine. These two lines are actually uneasy to understand, but when uh, uh, we are learning the principle of linkage, when we will learn the principles, when we will learn the experiment of Morgan on Drosophila melanogaster, we will easily understand these two lines later on into the next lecture. So here I am concluding myself with the chromosomal theory of inheritance, and that's the experimental proof which is given by T.H. Morgan, and theory is linkage and recombination. Once again, linkage is a group of gene which is always tries to inherited without getting changed or separated. They are going or inherited into the group from one generation to the other, that are showing the linkage. Recombination, they are getting separated, right? So linkage and recombination is one of the important principle which is proven by T.H. Morgan in order to uh, experimental proof of the chromosomal theory of inheritance. The basic two or three principles what we have seen earlier in standard 11 during the process of meiosis. One principle of G1 as G2 and rest of the meiosis 1 and 2, that was just now we have seen in Sutton and Bovary experiment. But independent assortment rule, what observed by Sutton and Bovary, could not be able to prove themselves. So none of the Sutton or Bovary could be able to explain why it is happening, why the interchange of the genes are happening. The very important word is uh, the change in characters. Why the change of the character which are seen, which are observed into the offspring? Why differences are seen? So the answer of that question, that tough question, is given by Morgan. He proposed the basic mechanism of variation, right? So variation is the changes of character of the offsprings, and how the variation is created, that is proven by T.H. Morgan in linkage and recombination experiments, which we are going to see into the next lecture. I'll see you into the next lecture, and all the best for understanding this, and thank you very much. I'll see you later on into the next lecture. All the best.